Successful achievements are always preceded by successful preparation. Prepare the umbrella before it rains. So preparation is the most important strategy for cracking any examination. So today here I am to talk about the important reference book and important topics on Unit 6 System Physiology plants. For your information, we have come up with a freebie which has important topics and mock test on Unit 6 System Physiology in plants. So please check out in the description box below, click onto the link, download and get benefited. With no more delay, let's get started to the video. Hey ardent learners, this is Caroline Green from Biotechnica. With no more delay, let's dive into this session. So first thing, we are going to talk about Unit 6 System Physiology in plants. So the first important thing that we are going to talk about in detail is what is going to be the reference book. When you start your preparation, it's really crucial that you know what are the books to follow. So these are the list of reference book that you can follow for Unit 6 System Physiology in plants. The first book, which is Plant Physiology by Lincoln Tays and Edward Ziegler. The second one is going to be the Plant Physiology by Rosin Salisbury. And the next book is Introductory Plant Biology by Stern. And the next one, Biology of Plants by Raven. And the Plant Biology by Egrios. And Plant Breeding Principle Methods by B.D. Singh. And the last one, if you want to have your instant notes created, then it is by Lack and Evans. So these are the list of books that you have to follow for Unit 6 System Physiology. If you really wanted to take a screenshot, you can take a screenshot of it. The next one. Okay, we already know about Unit 6 System Physiology Plant Syllabus. So this chapter is really crucial because you will be having 35 to 40 questions in this unit. So this is going to be the syllabus of Unit 6. Let's see into it. The first one is going to be photosynthesis. Photosynthesis is a very nice, easy topic that you can have direct questions from. So it's a promising chapter, I can say, or we can say it's going to be a scoring chapter. So in this chapter, we'll be talking about light reactions, we'll be talking about dark reaction, what are the light harvesting complexes, C3, C4, C2 pathways, everything in detail along with CAM pathway. And the next we'll be talking about during photosynthesis, the plant is actually going to produce glucose, and how is this glucose is going to be utilized by the plants for its own metabolism or to generate energy. That is going to be the second subdivision, we can say, which is going to be respiration. We'll be talking in detail about it. We'll be talking about glycolysis, citric acid cycle. We'll be talking about electron transport chain, oxidative phosphorylation in detail, and we'll also be talking about alternate oxidases and photorespiratory pathways in detail. The third one is going to be nitrogen metabolism, where we'll be talking about how an atmospheric nitrogen is fixed in the soil for the growth of the plant. What are the organisms that is involved in fixing these atmospheric nitrogen? We are going to talk in detail in this subunit. Next one is going to be plant hormone. How does a plant actually grows? What are the hormones that makes them to enlarge the stem or for the root formation or for the stem formation? So what are those plant hormones are going to be? So we are going to talk in detail about it, and how are they going to be helpful for the growth of the plant? We are going to talk in detail about it. The next one is going to be sensory photobiology. So here we'll be talking about certain mechanism of action of phytochromes, and we'll be talking about important stomatal movement and photoperiodism, which is an important one, and some biological aspects we are going to talk in this unit. The next one, you can see it over here. Solute transport. Yes. Suppose if the plant wants water or if the plant wants food, how this is actually been transported to the entire part of the plant. Suppose if we are talking about a plant, if a plant is very taller, but the water and the minerals are available in the soil, how this has been transported from the bottommost and till the tip of the plant, till the apex we can say. So how the transport mechanism is actually taking place. Here we'll be talking about water potentials, osmotic pressure, pressure potential, all these things in detail. We'll be talking about how does it gets transported, how the phloem loading and unloading is going to take place. Everything in detail we are going to study in subunit. The next one is going to be secondary metabolites. What are the plants that are going to produce some secondary metabolites, which is responsible or it's useful for human mankind, how it has actually been produced, what are their roles, everything we'll be talking. We'll be talking about terpenes and we'll be talking about phenols, everything in detail in this unit. The next one is stress biology. Yes, so stress, you have to remember stress hormone is going to be abscisic acid. So this is about stress physiology, where we'll be talking about the response of plants. How does a plant respond to biotic, or we can say living organism, if there's going to be any pathogens, or we can say some bacterial infections or viral infection, how a plant actually respond to that. Or if there's going to be any abiotic factor, which can be a non-living factors, like if there is less amount of water, maximum amount of water, less amount of light, or if it's going to be maximum amount of temperature and maximum salinity conditions, and how the plant is actually going to do this action. So this unit 6 is a very vast chapter, but it has direct questions in this chapter. Really important chapter, which you'll be having some 35 to 40 questions in this unit. So very important. Let's dive in and ask and talk about the important topics in unit 6. First, topic 1, which is going to be photosynthesis. Very important, very easy one, which we might be studying from our school days. So in this, we'll be talking about how actually the plant is going to produce glucose, which is nothing but a food. So it's actually a single step reaction we used to write, but we'll be talking what are the entire complete process that actually takes place over there. The first thing, we already know about light reactions. So light reaction is called as photophosphorylations. So here we'll be talking about what is a non-cyclic light reaction of photophosphorylations or a cyclic photophosphorylation. In this pathway, there might be question how many amount of ATPs and ADPH are going to be produced and how many are going to be utilized for production of glucoses. So here we know that if it's going to be non-cyclic photophosphorylation, there is a production of ATP and there's a production of NADPH. And please do not confuse with NADPH and NADH. NADPH is responsible for biosynthetic pathway. Synthetic pathway. So in non-cyclic photophosphorylation, there is a production of ATP and NADPH, but in case of cyclic photophosphorylation in stroma lamellae, it's going to be only ATP. So I'm just giving an overview. We are not dealing in detail. I'm just telling what are the important topics in this unit. The next one is going to be C3 pathway, or we can call it dark reaction. Dark reaction. Or we can otherwise call it Calvin cycle. So we can also call it Calvin cycle. And this is the most important biosynthetic pathway. In this pathway, what the plant is going to do, the plant is actually going to convert all the carbon dioxide that it has taken from the atmosphere. So we can write. So what they're going to do is, let me write it over here. So they're going to fix all this carbon dioxide into glucose moieties. Into a glucose moiety H12O6 by utilizing all the ATP and NADPH that it has produced in the light reaction. So it's going to be a light independent reactions, we can say, but it's always dependent on the products of the light reaction. So by utilizing ATP and by utilizing NADPH, they are going to produce glucose molecule, which is important for the plant. So this is the primary productivity, we can say, which the plant actually produces in large tons, we can say. So this is going to happen. So question can come like, how many molecules of ATP and how many molecules of NADPH are utilized for the production of one molecule of glucose, or how many ever molecules of glucose can come in your examination. The next pathway is C4 pathway. There are some plants, they do not want to go in for a reaction called as photorespiration. So in this photorespiration, what exactly happens is the plants, instead of taking the carbon dioxide, they try to use the oxygen, which is actually not a good process. We can say it's a wasteful process. So in order to prevent, or in order to not take that pathway, they go in for an other pathway, which is called a C4 pathway, where there are certain plants like sugarcane, sorghum. All these plants will undergo, instead of going for C3, they go in for C4 pathway. So this pathway is otherwise called as Hatch-Lack pathway. Hatch-Slack pathway. So in this Hatch-Slack pathway, we'll be talking about the bundle sheets and the mesophyll cells together. There's a role of these location where exactly this is going to happen, where you'll be studying in your classes. I'm just giving you an overview. And the next one is crassulacean acid metabolism. This usually takes place in an area where there is going to be less amount of water. We can say it takes place in area like arid areas, desert areas. Succulent plants usually undergoes this pathway. So there might be a question like they can ask you, there would be a plant which can actually undergo C3 pathway when water is available, but if water is not available or when they're going through a stress conditions like drought conditions, they will adapt to CAM pathway. So which of the following can be those things? So it's going to be the ice plant, we can say. So crystalline, so this kind of questions will come in your CSIR NET examination. So please make sure to read all the examples very importantly. The next is going to be photorespiration. We can correlate photorespirations in photosynthesis along with respiration also. There are certain plants which cannot bear high temperature, or instead this rubisco which is present will be acting as oxygenase and they will fix oxygen instead of fixing carbon dioxide. Why? Because the carbon dioxide become less and the oxygen content increases. So because of this, photorespiration will take place. So we are going to talk important thing in this one. The next, what are the enzymes involved in all these pathway? When you're going to read any pathway, it's really important to read all the enzymes, its role. If it's not going to be present, what's going to happen? So very important question we can say is rubisco. So what's the role of rubisco in C3 plant? Where is the rubisco present in C3 plant? Where is the rubisco present in C4 plant? So this is a very important one. And what is this rubisco is going to convert one molecule into an other molecule? That is also another important thing. Next one. Enzymes are there, but there has to be an inhibitor, right? Yes. So what are the inhibitors that actually prevent the pathway to take place? In plants, this can also happen, right? So you have to remember what are the inhibitors that halts or prevents all these pathway to take place. So these are some of the important topics that you have to study in your topic 1, which is going to be photosynthesis. Okay, the next one, respiration. Very lovely one. This is actually going to be because there's a correlation of photosynthesis and a respiration. We know that a plant has produced glucose. But anybody, suppose if I'm there, I need glucose for my own metabolism. The same way, plant is actually producers. They are the major producers. So they are the one which supplies glucose to you in the form of starch. So they have produced a lot of glucose here. How are they going to utilize this glucose? So glucose has to be converted into an energy currency, which is like a cash for them. So they are going to be converted into ATP. And now you can see it over here. It's not NADPH. It's going to be NADH. And ATP is going to be the energy currency. And this is going to be like a check. This is like a check, we can say. So if you have cash in your hand, you actually go to a mall, you purchase something else. But if it's going to be a check where you have to go to a bank, give it to them, draw the money, and then you purchase something, right? The same way here. So they can produce a direct energy, or they can either produce NADH and it's not NADPH over here. So in this, what exactly is going to happen? There are 4 important mechanisms you have to study in detail. The one is going to be glycolysis. Very easy. Lysis means breaking. So glycolysis, what's going to happen? We know that this glucose, which is going to be a 6 carbon, is going to be broken into a 3 carbon, which is going to be a pyruvic acid. So what's going to happen? When you break a bond, you actually get energy. So that's what it's going to happen in case of glycolysis. Whatever the plant has produced, now they're going to break it into energy. So what you have to study? You have to study each and every step in glycolysis. What's going to be the rate limiting step? What's going to be the enzymes? What are going to be the inhibitors which prevents this pathway? So you have to study rate limiting steps, 3 rate limiting steps, all the enzymes involved in it, and how have they been regulated? That's very important. The next one is going to be, let's talk in detail about TCA cycle, or we can say Krebs cycle, or we can say citric acid cycle. Krebs cycle, or we can say citric acid cycle. So this takes place in mitochondrial matrix, and here also you have to study all the enzymes involved in it, very importantly about succinate dehydrogenase, which actually involved in ETC mechanism also, which actually involved in this TCA pathway also. So you have to study this in detail about it. And the next comes the electron transport chain. Very interesting pathway if you understand this. So there are going to be 4 complexes which will be involving in it, and you have to study what are the complexes, what are going to be the ions that are actually present inside it, and you have to study what are the inhibitors that prevents this entire pathway to take place. So ETC we already know, where whatever we have this NADH, which is the indirect source of energy, will be converted into ATP molecules in this pathway along with oxidative phosphorylation. So oxidative phosphorylation, you have to know the location of it, F0, F1 particles. Very importantly, you have to study about what are the inhibitors that are actually present here. So major simple thing you have to understand is photosynthesis, production of glucose over here. You have to study everything in detail with a flow chart, clear understanding, and then you can go in for a respiration, how this glucose has actually been converted into ATP and NADH, and finally all the NADH and all the FADH will be converted into ATP. So study every pathway with a clear understanding, so it will be really helpful. So these are the important things that you have to study in topic number 2, respiration. The next topic, yes, the topic number 3 is we are going to see nitrogen cycle. Yes, there's a beautiful image which is given here, how the nitrogen which is present in the atmosphere has been fixed in the soil, utilized by the plants for its own growth. Yes, so there are Annabella, Nostoc. You have to study all the organisms name along with what are they going to convert it. So first important thing, nitrogen cycle. How is the nitrogen cycle actually takes place? So this is, you won't get that many questions over here, but you will get questions based on the enzymes, what are they going to be converted, what are the organisms that are involved in it. All these things are really, really important. And the next one, steps involved in biological nitrogen fixation. This is where the organism name actually comes. So how the nitrogen is actually fixed in the soil, because the atmospheric nitrogen is present over there in N2 form. So it has to be converted into usable forms. So how the bacterium or the organisms are involved in converting this. So you should remember all the organism names like nitrifying bacteria, denitrifying bacteria, everything together, and all the steps which you will get in your examination. The next one, root nodule formation. Yes, this is a very important question. It also has a correlation with the hormones. So auxins and cytokinins are certain hormones which are responsible. We can say auxins are responsible for root initiations, and cytokinins are responsible for shoot initiations. So these 2 hormones are actually responsible for the formation of a crown gall diseases. So agrobacterium tumefaciens we'll be talking about. So you have to remember that one also. And root nodule formation, how a nodule is actually formed in the root. That is mainly because of the rhizobium bacterium, and this rhizobium bacterium will always has an enzyme called as nitrogenase. We'll be talking all these enzymes later. So how is this enzyme actually work? This enzyme actually works when there is no oxygen available in the plant. And who's going to be the oxygen scavenger? That is an important question. So they'll always ask you questions like leguminous plants. This is a question which usually comes in CSIR NET. Leguminous plants will have less concentration of oxygen because of, there would be an option. So you have to remember it's because of leg hemoglobin, which is going to be an oxygen scavenger, and they will remove all the oxygen which is present over there. Then only the rhizobium can produce its enzyme and act upon and fix the nitrogen over there. So this is how they are actually forming a root nodule formation. So please remember this and understand all the organism, whether they are converting this nitrogen into ammonia, what is the enzymes that is responsible for reduction of nitrogen into ammonia. So you have to remember nitrogenase, and you also have to remember what are the metals that are actually involved in it. So they can ask you questions on which of the metal is not involved in it. So you have to remember whether it's molybdenum or Fe or vanadium. Cobalt is not involved in it. So you should remember all these things in detail when you are going to study this. Yes, okay. The next one is going to be topic 4. The next topic, phytohormones, or we can say it's going to be plant hormones. So this is a very easy one which you have to study in detail about it, because you would be confused over here. So remember all the phytohormones which is given in our syllabus. So which can be auxins, I told you. Auxins, you have to remember some synthetic auxins along with the precursors, all these things. And you have to remember cytokinins. What's the role of cytokinins? And what's the role of ethylene for fruit ripening and ABA? The stress hormone. What's the role of the stress hormone? And even we can say gibberellic acids. Gibberellins, we can say. I'm going to write it over here. Gibberellic acid, they can ask you questions like which of the following is going to be not the bioactive form of a gibberellin. So they can give you like GA1, GA3, GA4, and they can ask you GA5. So you can answer it like GA5 is not the bioactive gibberellin. So you can have questions on this plant hormones, or we can say phytohormones. The next one, very important, you have to study the receptors. Very important, you have to study the receptors and where is the location? Where is this hormone synthesized? And where is this hormone is going to act upon? That's very important. So what's the function of all these hormones is the next important thing. And the biosynthetic pathway. How are they actually synthesized? And their action, I told you the mechanism of action, they might be synthesized in one location, but the action is going to be in a different location. So you have to remember the mechanism of action. Suppose if the hormone is secreted in a higher amount, we can say hypersecretion of hormone, what's going to happen? Suppose if they're going to reduce, or if the production is going to be very less, or we can say hyposecretion, what are the situation is going to happen and their physiological roles. So study all these things with a clear understanding, so it's going to be very easy and you will get direct questions from this unit. Yes. Next one, topic 5, which is sensory photobiology. Yes. So sensory photobiology, which we can say phytochromes. We know chromes are going to be colors. So phytochromes, there are going to be cryptochromes. And phototrophins, there would be a question, and you'll be talking about transpiration. So there would be a question, the activity of the phytochromes will be done with which of these following? So you can answer the conversion of PR into PFR through the red light. So it's going to be very simple question, direct questions only. So you can study this. And then the stomatal movement, which is the closing and the opening of the stomata, when this is going to take place. So if the plant is going to have a very less amount of water, there's going to be a stress conditions for them. That time they don't want the transpiration to take place. So there's going to be a hormone, which is ABA ultimately. So when do they close? So you have to study about the opening and the closing of stomata and how actually the stomatal movement actually takes place. And what are the factors that actually responsible for these opening and the closing of stomata is also another important one. Yes. Next one, photoperiodism. Yes, some plants need more light, some plants need less light. What happens if a plant which requires more light is kept in a very less light area? So that you have to study in detail. So photoperiodism, which is long day and short length time period, you have to study in detail. The next topic is topic number 6. Here you can say, okay, solute transport and photoassimilate translocation. I already told you how are they been going, all the minerals, or we can say water or food has been transported. So you have to study in detail about how the solute has actually been transported, how they've been translocated. Everything you have to study in detail, where you'll be studying about your diffusion, osmosis, active, passive transport, everything in detail. The next one, topic 7 and 8 together I've given. So this is very important. The water potentials, which is going to be osmotic potential, or we can say water potential, osmotic potential, pressure potential. All these things are really important, and you have to understand what will happen if it's going to increase or it's going to decrease. That's a really promising one. The next one is ABC model, which you can see Arabidopsis, which is going to be the homeotic genes. This is really important. I'm telling you, this is really important. Make it a star point if you are listening to my video. So please make a star point. This is a very important one, which is ABC model in Arabidopsis. So we'll be talking about what are the genes. Direct questions will come from this. So please stick on to this one. Yes. Okay. The next one, topic number 9. Yes. The next one is going to be secondary metabolites. We'll be already talking about terpenes, right? Yes. So there can be questions on terpenes, phenols, and how their pathway actually take place and how they been synthesized is going to be some amount of things that you will get in this topic. Yes. The next one is the final one, topic number 10, stress physiology. Yes, you have to remember how does the plant is going to respond to its biotic and abiotic factors. So responses of plants to biotic factors, like what will happen if a pathogen actually comes and infects a plant, and then what happens if water increases or decreases, or the temperature increases or decreases, or if the saline condition increases or decreases, how the plant is actually going to face the stress. So it's going to be stress physiology, and plants detailed mechanism you have to study of how do they adapt themselves to all the stress conditions. So this is a very important thing. You should remember detailed mechanism, how they actually takes place, how do they adapt themselves to all this stress condition is going to be the next one. So I think we have done all the important topics in unit number 6, which is going to be system physiology in plants. So when you're revising or when you're studying, when you are going to start this chapter, it's really important to know all the important reference book. Along with that, you need to know what are the important chunk that you have in this chapter, which you should never ever miss. So I've told all the things in detail about it, and final note I'm going to tell you, yes, never underestimate the power of a planted seed. Yes, if you plant a seed and if you have water, if you're pouring minerals, everything together and provide fertilizers to it, we know it can become a giant tree. The same way you are, never underestimate yourself. Always do your best. So always do your best. What you plant now, what you are going to do it now, you will going to harvest later. So with this note, I'm going to end the session for today. Thank you all of you for joining, and thank you so much. And as I already told you in the beginning, there is a freebie which is given, which has all the important topics PDF and the mock test of unit 6 system physiology given in the description box below. So you can download, check out, download and get benefited. Thank you all of you for joining. We'll meet in the next video. Thank you all of you.